Hello students, in previous part we have studied about meiosis 1. Meiosis 1 is followed by meiosis 2. Meiosis 2 is also called as homotypic division. Now in between the meiosis 1 and meiosis 2 there is a short resting period or there is a short resting phase. That phase is called as interkinesis. The short resting phase in between meiosis 1 and meiosis 2 is called as interkinesis. Now this meiosis 2, in this process what happens? The haploid cells which were formed in meiosis 1, the two haploid cells which were formed in meiosis 1, those cells they further divide to form 4 haploid cells. And this division is similar to mitosis. The daughter cells which are formed in meiosis 2 are similar to their parent cells formed in meiosis 1 with respect to the number of chromosomes. And that is why this meiosis 2 is called as homotypic division. Means the number of chromosome in the parent cell, the cells which were formed in meiosis 1, those cells and the cells which are formed after meiosis 2, those 4 haploid cells, the number of chromosome in both the parent and the daughter cells are similar. And that's why this meiosis 2 is called as homotypic division. The parent cells and daughter cells are similar to each other with respect to the number of chromosomes. Now in this meiosis 2 there are 4 phases just like mitosis: prophase 2, metaphase 2, anaphase 2 and telophase 2. Telophase 2 is then followed by cytokinesis 2. Now in prophase 2, what happens? The nuclear membrane, as you can see in the diagram, the nuclear membrane and the nucleolus, they start disappearing. They start disappearing and the chromatin material which was there inside the cell, that chromatin material starts coiling together again. It starts coiling itself and it forms chromatids, sister chromatids. And after that again it condenses to form chromosomes. The centrioles which were there, they start moving to the opposite poles again and they give rise to aster rays, and both the centrioles are connected by the spindle fibers. In the second cell also same thing happens. The nuclear membrane and the nucleolus, it starts disappearing. The chromatin material, the elongated chromatin material which was there, it starts condensing and it starts coiling together to form chromosome. The centrioles starts moving away from each other to the opposite poles. They give rise to aster rays and both the centrioles are connected together by spindle fibers. So the prophase 2 is followed by the metaphase 2. In metaphase 2 what happens? The nuclear membrane and the nucleolus completely disappears and the centrioles, they move towards the opposite poles. They have moved towards the opposite poles and they are connected by the spindle fibers. Some of the spindle fibers, they are attached to the centromeres of the chromosomes. Now these spindle fibers, they are attached to the centromeres of the chromosome, and now what happens? The chromosomes are then arranged in an equatorial plane. They are arranged in the middle, in the equatorial plane, and the spindle fibers, they pull those chromosomes towards the opposite poles. These spindle fibers, they start pushing and pulling the chromosomes towards the opposite poles. This pole will pull the chromosome downwards, this pole will pull the chromosome upwards. So they start pushing, they start pulling the chromosomes towards the opposite poles. And in the second cell also same thing happens. The nuclear membrane and the nucleolus, they disappear completely. The centrioles, they move towards the opposite pole. They are connected with the spindle fibers. Some of the spindle fibers, they are attached to the chromosome by their centromere, to the centromere. They attach the chromosome to the centromere where the chromosomes or the strands of chromosomes are joined together. And the chromosomes are arranged in an equatorial plane and the spindle fibers, they start pulling the chromosome at the opposite poles. So the metaphase 2 is then followed by anaphase 2. Anaphase 2 is the shortest stage in meiosis 2. In anaphase 2, what happens exactly? The spindle fibers which were attached to the chromosomes, those spindle fibers, they pull those chromosomes towards the opposite poles. As due to this pulling action, the centromere of the chromosome breaks down into two or gets divided into two, and the chromosomes separate from each other and they are pulled towards the opposite poles. As the chromosomes separate from each other, the daughter chromosomes are formed. The separated chromosomes, those are called as the daughter chromosomes. Daughter chromosomes are formed, and those daughter chromosomes are then pulled at the opposite poles by the chromosomal fibers. In the another cell also same thing happens. The chromosomes which were attached to the spindle fibers, those spindle fibers, they start pulling the chromosome. Due to this pulling action, the centromere to which those chromosomes were attached, to which those strands of chromosomes were attached, that centromere gets divided into two and the chromosomes become separate. And as they become separate, they are called as daughter chromosomes. Daughter chromosomes are formed, and these daughter chromosomes are then pulled by the chromosomal fibers towards the opposite poles. And now the anaphase is followed by the telophase. During this stage, what happens? The daughter chromatids which are formed, those daughter chromatids, they start uncoiling themselves and they again become elongated to form chromatin material. The daughter chromatids uncoil themselves and become elongated to form chromatin material. And the nuclear membrane and the nucleolus, they start to reappear. In the other cell also same thing happens. The daughter chromatids uncoil themselves to form chromatin material. The nuclear membrane and the nucleolus, they reappear. So here the end of the telophase indicates that the end of karyokinesis. After this, the end of the telophase is then followed by the cytokinesis too. After telophase, the cytokinesis takes place. In cytokinesis, the cells, the haploid cells, they further divide into two haploid cells. One haploid cell divides into two haploid cells. Thus, the four haploid daughter cells are born or they are formed. Now here, cytokinesis, as we know, it is the division of cytoplasm. So cytoplasm starts to constrict and it constricts deeper and deeper. That results in the formation of two new daughter cells. In the same way, the other cell also, the cytoplasm of it constricts more and more, and this results in formation of two new daughter cells. So in all, four new haploid daughter cells are formed. And now as for the significance of meiosis, that is, it produces haploid gametes for sexual reproduction. Half number of chromosomes is present in the daughter cells. That means the daughter cells, they have the number of chromosomes half to their parents. It maintains constant number of chromosomes. The number of chromosomes is maintained to constant. Genetic recombination due to the crossing over. Due to the crossing over, the genetic material is exchanged in between both the chromosomes. So that is genetic recombination. And this leads to the variation and evolution. That this genetic exchange of genetic material, it leads to the variation in an organism or variation in a human being, or it leads to evolution, or it gives rise to evolution. Now what is crossing over? As you can see this diagram over here, these are the homologous chromosomes, these are the sister chromatids, these two are the non-sister chromatids because they are from different chromosomes, and the point at which the chromosomes are attached is the centromere. Now these homologous chromosomes, they cross over each other at a point called as chiasmata, and at this point is called as the point of crossing over. After crossing over, as the genetic material gets exchanged, the chromosomes, they start repelling each other. The repulsion takes place, and after the repulsion, still those chromosomes are attached to the tip, attached by the tip, that is the terminalization. they are terminalized, or they are attached to the terminals. That's why it is called as terminalization. And after terminalization, the repulsion takes place, and finally the chromosomes, they get separated, and these are called as crossed over chromosomes because the genetic material is exchanged. So this is the crossing over.